Preparation model paper 30. I'm Shankar Reddy, PhD plant pathology. 301. Heterogeneous demicyclic rust. Just an example of heterogeneous demicyclic rust. I think the question number 300 is an example of autogeneous demicyclic rust, probably rubus orange rust and white pine blister rust. And in continuation 301, heterogeneous demicyclic rust. The best example is we can call it as the car cedar apple rust. That is the best example for heterogeneous demicyclic rust. Generally, what is demicyclic? Lack of iridial stage is known as demicyclic. So the next one is the number of germ pores produced by the iridospores. So the iridospore generally produces 4 number of germ pores. The number of germ pores, like in wheat stem rust, there are 5 kinds of spore stages are there. Stage 0, stage 1, stage 2, stage 3, stage 4, stage 5. That is pycniospore, achiospore, iridospore, teleospore, and bestiospore. So like for each and every spore have different kinds of germ pores. Here this iridospore produces 4. So the number of germ pores that is produced by this, especially the rust spores, the different kinds of germ pores that is produced by the rust spores that we have mentioned in the mycology section. Generally the iridospore produces 4 number of germ pores. The next one is potato scab potato scab severe in soil at what pH? So at the rate of pH 8.0, the potato scab is very severe. When we drop down the pH to around 5.5, we can reduce the disease. And in 1845, lead blight of potato is due to a fungal pathogen called Phytophthora infestans. In 1943, it's not 43, it's 42. In 1942, Bengal famine, 42-43 actually generally, Bengal famine is caused by Helminthosporium oryzae. The next one is father of modern plant pathology. Anton de Berry is generally considered as a father of modern plant pathology, father of experimental plant pathology. All those things are related to Anton de Berry only, because he have done lots of greatest contribution to the timeline of plant pathology in the development of that what we are doing now. So, and he also the one who discovered the life cycle of lead blight of potato and came to a conclusion this lead blight of potato is a fungal pathogen, and he also gave this Phytophthora infestans. In Phytophthora, phyto means plant, thora means destroyer. Gene to gene concept or gene for gene hypothesis in 1946 was given by, so gene for gene hypothesis was given by H.H. Flor. So this is a very important concept that actually just changed the way of thinking and perception towards the plant diseases. So the concept was actually given by H.H. Flor, founder of virology. So the term virus was actually coined by Bijernich, and is also the person who gave the meaning of the virus, which is called which now we know as poison or venom or slimy liquid. And the TMV, tobacco mosaic virus, that is mosaic disease of tobacco, was first caused by tobacco mosaic virus. That is also told by this Bijernich only. He is considered as a founder and father of plant virology, the first Indian scientist who collected and identified the fungi from India. So K.R. Critiker in 1885, he is an Indian scientist. Before Cunningham or Sinningham in 1883 and 82, he is also there a scientist from European countries. He is there in India, used to collect the different kinds of fungi, but he is not an Indian. When it comes to this K.R. Critiker, he is an Indian who started the collecting fungi in India at 1885. Fungi and diseases in plants was written by E.J. Butler. Fungi and plant diseases was written by B.B. Munkur. I repeat, fungi and diseases in plants was written by E.J. Butler. Fungi and plant diseases was written by B.B. Munkur. So this E.J. Butler wrote in the year of 1918, whereas B.B. Munkur 1948. So we have discussed this heterogeneous, something like that, right? So these are all the overall list of this autogeneous macrocyclic rust. Example of autogeneous macrocyclic rust. So BFSs like bean rust, flaxseed rust, sunflower rust, safflower rust, castor rust, all are the examples of autogeneous macrocyclic rust. So bean rust is caused by Euromyces appendiculatus, flax rust is caused by Melanospora linea, sunflower rust is caused by Puccinia helianthus, safflower rust is caused by Puccinia carthami, castor plaster rust is caused by malums or a ricin. So these are all the different kinds of examples that comes under this autoecious macrocyclic rust. Autoecious demicyclic rust means rubus orange rust. So the rubus orange rust is the best example for autoecious demicyclic rust. The next one is heteroecious macrocyclic rust. Heteroecious macrocyclic rust, best example is wheat stem rust and white pine blister rust. Wheat stem rust is Puccinia gramineaceae. This is the best example, and white pine blister rust, that is Cronatium rubicola. And heteroecious demicyclic rust, just now in the 301, the question number 301 we have saw, right? So this is CAR, cedar apple rust, Gymnosporangium juniperi virizinane. Alternate host for cedar apple rust, the alternate host for cedar apple rust is apple only. So the lack of demicyclic rust is known as iridial stage. Sorry, if the iridial stage is absent, it is known as the demicyclic rust. So which stage is absent in demicyclic rust? Iridial stage is absent. Best example is cedar apple rust and rubus orange rust is the best examples, where examples of this demicyclic rust. In both the cedar apple rust and rubus orange rust, iridial stage is absent. Fungi and diseases in plants. I think the previous question only, fungi and diseases in plants was written by E.J. Butler. Fungi and plant disease, now it came here. Fungi and plant disease was written by B.B. Munkur. Bacterial variability can be achieved through. So bacterial variability can be achieved through three kinds of mechanism: conjugation, transformation, transduction, right? So in asexual reproduction, variability cannot be achieved. Same kind of, if you see budding, same kind of bacteria will be produced. Even fission also same kind of, but only a sexual reproduction kind of thing only we can generate some kind of variability. We can't call as a bacterial sexual reproduction. Instead of that, we can call as bacterial recombination. So there are three kinds of recombinations are there in bacteria, that is a conjugation, transformation, transduction. So these are all the three kinds of bacterial bacterial sexual reproduction or sexual variability mechanisms that we can see. So generally variability can be created by sexual reproduction. The sexual reproduction types is or sexual recombination types in bacteria is conjugation, transformation and transduction. JC Luthra solar heat treatment is applicable for. So JC Luthra where solar heat treatment is applicable there. This is called wheat loose nut Stulagonoda. Who did most of his work on rust disease in India? In India is a scientist called K.C. Mehta. He is also called as father of rust, Indian rust generally. Okay, is K.C. Mehta have done tremendous amount of work, especially on the rust disease in India. Common scab of potato controlled by reducing the pH. At what pH we can control this common scab of potato? If we drop down the pH at 5.2, because the disease is severe at 8.0. Okay, the disease is severe at 8.0. If we bring down the soil pH to 5.2, we can manage the disease. And there is another case called clubroot of cabbage. Clubroot of cabbage is severe at 5.2 to 5.5, and we can control the at the pH of 7.6. If we raise the soil pH by the application of lime, we can manage the disease. And the next one is a very important disease caused by mycoplasma like important disease caused by mycoplasma like organisms. There are lots of diseases are there. You can take mulberry dwarf, little leaf of brinjal, sesame pillodi, and all are the most important diseases in India. In India, we can say sesame pillodi and little leaf of brinjal. It's my personal experience. I have many brinjal fields. I have seen this little leaf of brinjal, especially Tamil Nadu, near the area of Salem, surrounding areas of Salem. This little leaf of brinjal is very severe. And the next one is father of plant virology in India. So generally father of plant virology, Bijernich. In India, there is a scientist called S.P. Rai Chowdhury. S.P. Rai Chowdhury is considered father of Indian plant virology. Father of Indian mycology. Father of Indian mycology and Indian plant pathology, E.J. Butler. E.J. Butler is generally considered as father of plant pathology and father of mycology in India. So green ear or downy mildew of pale millet was first reported in India by E.J. Butler. In the year of 1913, I probably is the one who reported this pale millet, downy mildew for the first time in India. And the next one is the downy mildew of pale millet is primarily, so the downy mildew of pale millet is primarily a soil-borne pathogen. So these are all the different kinds of books that was on the scientists who wrote it. Gaspard Bohin, which wrote Phenoxyetry Botanici. In previously we have mentioned two books, right? Fungi and Disease in Plants or Fungi and Plant Disease. There are other couple of important books here. Gaspard Boyan wrote Phoenix Theatre Botanist in 1623. P.M. Micheli wrote a book called Nova Plantarum Genera 1729. Linnaeus wrote a book called Species Plantarum 1753. And C.H. Parsons Synopsis Methodica Fungorum 1801. E.M. Fry's Systema Mycologicum 1821. J.G. Kuhn. This is the first book that is completely devoted to plant pathology. So all the other books are related to taxonomy, classification and all the other things. But this book, which was written by J.J. Kuhn, is completely devoted to plant pathology. The book is Diseases of Cultivated Crops and Their Causes and Their Control, which was published in the year 1858. And this book is considered to be the first book that appears especially related to plant pathology, and that is related to plant diseases. Okay, Charles Darwin wrote a book called Origin of Species 1858 and 59. Anton de Berry wrote a book called Comparative Morphology and Biology of the Fungi, Mycetosia and Bacteria. Actually, the German version published in 1866, but the English version that was translated by Bayflor and one more man is there. Bayflor and something will be there. They actually translated Bayflor and one more person. They translated this German version into English in 1887. And Tulsen brothers wrote a book called Selecta Fungolum Carpologica 61 to 65. Secado produces a book called Silum Fungorum. It's almost a 25 to 26 volumes of massive book published in between the years of 1880 to 1931. E.J. Butler, Fungi and Disease in Plants 1918. M.J. Tirumalacher, Antibiotics and Plant Disease Control. B.B. Munkur, Fungi and Plant Disease 1948. Y.L. Nina and P. N. Thapliyal, Fungicides and Plant Disease Control 1971. Paul Niergaard wrote two books, Seed Pathology 1977 only. The first volume is completely subject to type, and second volume is only the references of the related to the first volume. The next one is the loose smut of wheat is generally they're asking whether the loose smut of internally seed borne or externally seed borne. The wheat loose smut is the best example for internally seed borne pathogen. Covered smut of barley is a externally seed borne pathogen. Loose smut of barley is a internally seed borne pathogen. Let me tell you, the loose smut of wheat and barley is the best example for internally seed borne pathogen, but the remaining majority of the plant pathogens are externally seed borne. Okay, and couple of other may be there, but the repeatedly asked question is this loose smut of wheat is a internally seed borne or externally seed borne? Internally seed borne. Okay, early blight of potato is caused by Alternaria solani and Alternaria alternate also. In India, wilt disease of sugarcane was the first reported in from Bihar probably. From Bihar, this was first reported, and the incidence of black scarp of potato was severe in which soil? Right, it is more severe in sandy soils. So the soils are sandy where you are growing the potato. This black scarp is very severe. Sheath blight of rice is more during the active tillering stage. This sheath blight is very severe. Example of silver lacking organisms like L-form of bacteria and a couple of bacteria where they don't have silver that belonging to this group of L-form. L-form means silver lacking bacteria. Okay. Father of microbiology, generally Anton von Leeuwenhoek is considered a father of microbiology, but father of modern microbiology is Louis Pasteur. And plant pathology. So generally in the name of plant pathology there are a couple of books, but generally the plant pathology book, this book was actually started by a man called G. Negrius, actually professor. Now the 6th edition is running up, 2023 August probably it's published in. And this edition was edited by Oliver, and the first 5 editions was written by G. Negrius only. After the G. Negrius after his last breath, and Oliver it took almost the 5th edition was published in 2005 probably, not probably 2005 only, and the present edition, the 6th edition, is running in 2023. See, almost 18 years gap, right? And James Oliver, probably Oliver is the one who edited this book. Now this plant pathology book, G. Negrius plant pathology book, is generally considered as Bible of plant pathology, which is a very fantastic book. In fact, there are soft copies also available in internet. Just you type G. Negrius plant pathology 5th edition, and there are plenty of soft copies available, probably 180 MB the size of the book is. And the 5th edition is around 922 pages, and the 6th edition is almost updated with 1100 pages. And in fact, I'm also waiting to purchase this book, but it's not available in India. Let's see in the upcoming days. Plant diseases in India was written by, plant disease in India was written by R.S. Singh. So he is also considered as a lighthouse of Indian plant pathologists and plant pathology. And MLO diseases are transmitted by. So MLO diseases are generally transmitted by leafhoppers. All phytoplasmal and spiroplasmal diseases are transmitted by leafhoppers. The majority of the phytoplasmal and spiroplasmal, but a couple of other means of spread is also can be observed, but majorly it is transmitted by leafhoppers only. In North India and in Central India, so the black rust of inoculum is actually produced from where, which means, so the inoculum for this black rust of wheat is generally producing from South India. It is believed to be Nilagiri Hills. Not probably, not exactly, but it is believed to be the South India. Colony morphology of Aspergillus tamari. Aspergillus tamari generally called morphology is a brown color. Alternate host for wheat leaf rust, Thalictrum species is considered to be as alternate host for this wheat leaf rust. Wheat stem rust is barberry, right? Single celled teleospore is produced by Uromyces, double celled Puccinia, multi-celled Phragmidium, parachute-like teleospore, Ravenelia. So these are all the types of teleospore that we have published in the mycology itself. Okay, crust-like teleospores is produced by Melonsora. Melonsora produces crust-like teleospore. And the sexual stages of the following: Penicillium, Thalaromyces, Graphium rosalina, I think, Monilinia ophiostoma, Dermatophora ceratocystis, probably Chalara, Monilina was a little bit, but Penicillium it's Thalaromyces only. Graphium, ophiostoma, ceratocystis, Graphium, I think, ceratocystis, Graphium. Monilina only, ceratocystis graphium, that is Dermatophora rosalina, I think, Dermatophora rosalina, Chalara ophiostoma probably. Please just go through once again. Penicillium is Thalaromyces, Dermatophora rosalina, Graphium ophiostoma probably, and Chalara ceratocystis or Monilina, oh, probably. So please go through this. I was a little bit confused about this. It's been many days, so just please go through this, or else I will comment in the comment section as well. Endospore staining agents. So malachite green is the one who generally recommended for endospore staining agent. What is endospore? We know very well. It is a heat-resistant spore or some kind of spore that is produced by the bacterial genus like Bacillus and Clostridium. The size of plasmid generally ranges from 200 kb. Here there are other staining agents I have given: endospore staining agent malachite green, plasmid staining agent acridine dye, capsule staining agent Indian ink, flagella staining agent carbol fission, lichen staining agent orchin, inclusion body staining agent azurea, cytoplasmic nucleus staining agent acetic acid, food storage organ, that is bacterial food storage organ. Bacterial food storage organ name is a polybeta hydroxybutyrate. We can also call it as a beta hydroxybutyrate. So that storage staining organ is a Sudan black bee. Yes, I have given the sexual stages here. Okay, Alternaria, Livia, Aspergillus, Eurotium, Sartoria, Emericella, Bipolaris or Helminthosporium, Cochleobolus, Botrytis, Botryotinia, Cercosporidium, Cercospora, Ramularia, Scocaita, Septoria, all are Mycospirilla and Didymella, Chalara, Ceratocystis, right? Then Cladosporium fulvia, Colrotrechum glomerella, Coniotherium leptospirea, Cryptospora leucostoma, Carvularia cochleobolus, Dermatophora rosalina. Yes, Dermatophora was there, rosalina. Okay, Dothiodorella diplodia, Botrospera, Dracellera pyranopora, Exerohylum septosparea, Glycladium trichoderma hypochrea rufa, Graphium ophiostoma, Marsonia, Diplocarpon, Monilia. Okay. Necator, Corticium or Pellicularia, Oidium ericife, Ophiobolus leptospirea, Faciliomyces bisoclamis, Penicillium uromyces and Thalaromyces. This was given Penicillium Thalaromyces, right? Phoma leptospirea, Phomopsis diaporte, Philastictinia grignordia, Poria felinis, and Rhizoctonia theratophorus. Again, Rhinoctoria. One more thing is Ceratobasidium, Rhizomorpha armillaria, Spisolima elsinoi. So those are all the various teleomorphic and anamorphic stages of the pathogen. And the next one is almond leaf scorch is caused by Xylella fastidiosa. I think in the previous lectures we have mentioned there are various number of diseases that is caused by this Xylella fastidiosa. I will tell you what are all they. Pierce disease of grape vine, phony peach called almond leaf scorch, citrus variegated chlorosis, and alfalfa dwarf, coffee berry, something, one more than there. All these diseases are caused by a bacterium called Xylella fastidiosa. Here the almond leaf scorch is also caused by Xylella fastidiosa. Name the pathogen that enters into the hydrothodes. Okay, there are two important pathogens that we used to study repeatedly: clubroot of cabbage, sorry, black rot of cabbage, black rot of cabbage. Black rot of cabbage and rice bacterial leaf blight. These two are generally considered to be the entrant guiders. Of course, other pathogens also there, but these two are very important. Okay, black rot of cabbage and rice bacterial leaf blight. The next one is name the pathogen that enters through trichomes. They're asking trichomes. Bacterial canker of tomato. Bacterial canker of tomato generally enters through trichomes. Clavibacter michiganensis, subspecies michiganensis. Agrobacterium was discovered in the sense genus was erected by Con, CON in 1942. Next one, Pseudomonas. Pseudomonas was erected by Migula, probably 94, I think. The largest plant virus family, 40 virus or 40 viridae is considered to be the largest. The longest in the length, the Clastrovirus, probably 1000 or 2000 nanometers, probably I'm not sure. The two citrus is the virus, probably. Citrus is the virus, I think so. The smallest viroid. There is a viroid called coconut kadang kadang. Kadang kadang means dying. Kadang means dying. Again kadang means dying. So coconut kadang kadang is reported to be the smallest, which consists of just 246 nucleotides. And the next one is a book written by Van der Planck. Actually, Van der Planck wrote so many books like plant disease epidemics and their control, year of 1963, and other books also there, right? Principles of fungicidal infection, host pathogen interaction, like that. He wrote 5 important books, but among that there is one book is very famous, that is a plant disease epidemics and control, which was published in there 1963. Puccinia pathway was identified by Nagarajan and Joshi. They identified the Puccinia pathway. And Indian stem rest rules was written by Nagarajan and Singh 1975. Puccinia pathway was identified by Nagarajan and Joshi 1980. These are all the entry points of important plant bacterial diseases. Hydathodes, black rot of cabbage and rice bacterial blight. So black rot of cabbage causes Xanthomonas campus, Pathovar campus. Bacterial blight of rice is Xanthomonas varese, Pathovar varese. Floral pods, that is nectar, is fire blight of apple and pear, which enters through Erwinia amylovara. Lenticels, potato common scab and soft rot of vegetables. So potato common scab is caused by Streptomyces cabis, whereas the soft rot of vegetables is caused by Erwinia kertovara. Trichomes, bacterial canker of tomato, which is caused by Clavibacter michiganensis, subspecies michiganensis, and through stomata there are a couple of pathogens like bacterial lipstick, citrus canker, angular spot of cotton. These are all the different kinds of pathogens that enters through stomata. So we may expect at least one question from this, because if you see the last previous just some 4 or 5 years question papers, there must be one question from this. So just please go through once this also. And other things also given here, I think. Insects, bacterial wilt of corn caused by Erwinia stivata, Pantovis stivati, which is transmitted by a corn flea beetle, Chitonema pulicaria, whereas bacterial wilt of cucumber is caused by Erwinia trachyphylla, but it is transmitted by a vector called typed cucumber beetle, Diabrotica victata, now name changed to Calama victata. Spotted cucumber beetle, one more beetle is there, that is Diabrotica duodecim punctata. Then root hairs, through root hairs, root nodule bacterium rhizobium, and through nematodes Whittier cockle, that generally caused by Rathibacter tritici, enters through a nematode called Anguna tritici. And one more disease called bacterial wilt of cornation, which is called Paraburkholderia caryophily. This enters through a root knot nematode called Melogain incognita. And the next one is the AUDPC developed by, first of all, we'll see what is AUDPC, area under disease progress. It's a kind of calculation technique to just observe this disease severity, all the stuff. This was first given by J. van der Planck. Okay, he is also considered as the father of plant disease epidemiology, J. van der Planck, right? And a simple interest disease and monocyclic. Simple interest disease, also known as monocyclic disease, produces a curve called saturation curve. Simple interest diseases or monocyclic disease produces a curve called saturation curve, whereas compound interest disease or polycyclic disease produce a curve called sigmoid curve or S-shaped curve, whereas polyetic disease produce a curve called bimodal curve. First computer simulation model, EPIDEM, recommended for early-blight potato and tomato, I think so. EPIDEM, okay. Van Everdingen rules or Dutch rules are applicable for lead blight of potato, Padabdor infestans, okay. Dutch rules are Van Everdingen rules, Irish rules, these are all are applicable for lead blight of potato only. Mills rules are applicable for apple scab, okay. Mills rules are applicable for apple scab, whereas the Van Everdingen rules, Dutch rules, Borke rules, Beeman rules, Irish rules, all these rules are applicable for lead blight of potato. Striga striate absorb nutrients through whatever the phaneromic plant parasite, irrespective, don't see the name. If it is a phanerogamic plant parasite, it absorbs nutrients through Hastoria. All phanerogamic plant parasite, whether it is a Striga, Asiatica, Striga, Katrina, whatever it is, if it is a phanerogamic plant parasite, is absorbing nutrients through Hastoria only. Just for example, okay. Jokes apart. Anyway, the next one is the pala blight of sugarcane is caused by. It's generally nutrient deficiency, manganese. So due to manganese deficiency, it will occur. Seed rules were passed in there. Seed rules were passed in there 1968. Insecticide rules were passed in there 1971. What is the DPPQ stands for? DPPQ stands for Directorate of Plant Protection, Quarantine and Storage, located at Faridabad. Started in the year 1946, I think. This DPPQS is the chief governing body where all this quarantine comes under. DPPQS, let me repeat once again, Directorate of Plant Protection, Quarantine and Storage, located at Faridabad, Haryana. And regarding the question number 358 and 359, on the right side I have given something. So 1912 Plant Quarantine Act in USA, that is also called as Federal Quarantine Act. The same act in India was passed with the other name, that is called DIP Act. Destructive Insect and Pest Act was passed in there 1914. It's a very important. DIP Act was passed in the year, or expanded DIP Act, Destructive Insect and Pest Act, which was passed in the year 1914. Okay, 1946 Directorate of Plant Quarantine Act. So where this DPPQS comes, Directorate of Plant Production, Quarantine and Storage. Okay, 1966 Seed Act, 1968 Insecticide Act, 1968 again Seed Rules, 1971 Insecticide Rules. These are very important. 1972 Federal Quarantine Pesticide Act, 83 Seed Control Order, 96 Food Quality Product Act, 2001 Protection of Plant Varieties and Farmers Right Act, and 2002 National Seed Policy. So these are all very important, especially this Seed Act, Insecticide Act, Seed Rules and Insecticide Rules are need to be considered more. ITCC stands for Indian Type Culture Collection, located in New Delhi. Composition of burgundy mixture. So generally there are some couple of copper fungicides are there, important copper fungicide. Bodo mixture, burgundy mixture. Burgundy mixture is also known as soda borax or mason lime. Why it is called mason lime? Because it was developed by a man called Mason in 1887. Okay, that's why it's called as mason lime. So burgundy mixture also known as soda borax. Generally the composition is 1 kilogram of copper sulfate, 1 kilogram of sodium carbonate, 100 liters of water. The main difference between Bodo mixture and burgundy mixture is, in Bodo mixture 1 kilogram of copper sulfate, 1 kilogram of lime, 100 liters of water. In burgundy mixture, instead of 1 kilogram of lime, we are using 1 kilogram of sodium carbonate. That is the only difference between Bodo mixture and burgundy mixture. The remaining is same. Fungicide act as a plant defense activator. There is a fungicide called metaloxyl. Not only metaloxyl, couple of other acebenzole, resmethyl, and all the fungicide. Among this plant, this metaloxyl is very important. This act as a plant defense activator. Who first used this Bodo mixture in India? Bodo mixture in India was first used by a man called Lawrence against Thicka disease, leaf spot, Thicka leaf spot of groundnut, I think, probably 1915, I think so. Metaloxyl was developed in there 1977. Okay, mode of action of benzimidazole. Generally, benzimidazole group of fungicide inhibits beta tubulin biosynthesis. Substrate used in DIBA. So dot immuno binding assay, I think so. BCIP, there is a substrate called BCIP, that is BCIP 5-bromo 4-chloro 3-indole phosphate. Or one more thing also can be used, nitroblue tetrazolium, that is generally called as NBT, nitroblue tetrazolium, that is generally can be recommended as substrate. DIBA plate is made up of, it is generally made up of nitrocellulose only. Okay, majority of the plates is nitrocellulose and other alkaline nitrocellulose and polynitrophenyl phosphate, something like that. Plates are generally made of, but this DIBA plate is made up of nitrocellulose membrane. Monograph on further studies on cereal rust in India was published by. So monograph on further studies on cereal rust in India first published by K.C. Mehta, where 2 volumes are there. Volume 1 which was published in volume 1 in the sense further studies on cereal rust, India 1, India 2. India 1 is published in 1940, India 2 published in 1952. But K.C. Mehta died in 1950 itself. Maybe probably the students helped in publishing in 1952 probably. East Genome Project happens in years between. East Genome Project is considered to be the first genome sequencing project in fungi. In plant, pathogenic fungi means that is rice blast only, Magnum porthugricea. In generally fungi means yeast. Okay, it happens in the year between 1989 to 1995. Vector for citrus greening. So citrus greening generally transmitted by a vector called Psyllids, Diaphorina citri. Okay, citrus exocartos is an indicator plant. Uh, Trifoliate orange, exocartos is trifoliate orangeole, I think so. Indicator plant for citrus disease virus. Citrus disease claxilla. Food storage organ for bacteria or bacterial food storage organ, a poly beta hydroxybutyrate. This question was asked in 2023. Okay, beta hydroxybutyrate or poly beta hydroxybutyrate or hydroxybutyrate, something like that they will ask. Okay, ISR, induced systemic resistance, is induced actually by the external application of chemicals. Right, what are the chemicals? So for the activation of SAR we can recommend salicylic acid. For the activation of ISR we can recommend jasmonic acid and ethylene. Rescue treatment fungicide, adimifon is generally considered a rescue treatment fungicide. Pronus necrotic ring spot virus is transmitted by pollen. It is also important question. I think in the previous question paper also we saw this question. Pronus necrotic ring spot virus is transmitted by pollen. Okay, enzymes involved in viral replication. So generally for virus replication, RNA polymerase 1 is required. For viroid replication, RNA polymerase 2 is required. Since they asked virus replication, for virus RNA polymerase 1, if it is a viroid, RNA polymerase 2. Father of soil solarization, Jacob Cotton. Jacob Cotton is generally considered as the father of soil solarization. He's an Israel scientist. Okay, and in fact I spoke to this, spoke in the sense I just mailed this mail regarding some doubts and clarifications regarding, and in fact he took some time and mailed me back. How, like that's a great experience for me. The next one is virus is a nucleoprotein, was given by Bowden and Pyre. There are two scientists where they called virus is a nucleoprotein. Why they called virus is a nucleoprotein, which means virus is a combination of nucleic acid and protein, right? So that's why they called as a nucleoprotein. Bowden and Pyre, 1936. Simple interested disease produces a curve called, so generally simple interested disease produces a curve called saturation curve. Okay, banana streak virus is transmitted by, generally banana streak virus and the cocosolin soot virus both are transmitted by mealybugs, Pseudococcus nigalensis. Apple proliferation is caused by Phytoplasma. TMV contains how many number of nucleotides? Generally this TMV contains 6400 nucleotides. Which bacteria produces a red color stain while adding to the milk? Generally there is a bacteria called Serratia marsans. It is a human pathogen as well. So if a red color pigment can be observed on the milk, that is the infection of Serratia marsans. If a green color pigment is observed on the milk, it is an infection of Pseudomonas aeruginosa. Trade name of pendimethalin. So pendimethalin stomp. Next one, monograph of Fusarium was written by C. Booth. C. Booth was the one who wrote this monograph on Fusarium, 1971. Life cycle of clubroot of cabbage was discovered by Oronin. Is also a student of Anton de Bary. He discovered the life cycle of clubroot of cabbage, and he also the one who named the pathogen as Plasmodium brassicae in 1876, I think. Example of autoecious macrocyclic rust. In the previous slides only we have saw the difference between various kinds of right. So the best example for autoecious macrocyclic rust is bean rust, flax rust, sunflower rust, safflower rust. All are the examples of autoecious macrocyclic rust. Number of germ pores produced by the teleospores. So teleospore generally produces 2, iridospore 4, like that will be there. For regarding all those things, please refer the mycology section that we have published in the previous year itself. Explain gene for gene concept in a single line. For every resistant gene in the host, there must be a corresponding and avirulence gene in the pathogen, and vice versa, like a continuous evolution process. Simply I will tell you it's all fight between the R gene and AVR gene, right? So dominance for upper hand, nothing else. So this concept was given by H.H. Floor, 1946, on flaxseed rust. Weight of bacteria can be measured in. So this is a tricky question, because previously everyone will thought bacteria is like a minute microorganism, maybe something else, but I don't know why this came up. Generally it is reported in many areas that gram, how we can measure the bacteria by gram. It's not at all you can put millions of microbes, that doesn't comes to close to gram also, that doesn't comes to close to gram, right? Then how they are measuring this weight of the bacteria in gram, I don't know. It's a little bit doubted about. If you have more information about this this measurement, please let us know. Give an example of Atrachus flagella. So regarding flagella, in the previous presentation we have given. Anyway, I'll tell you just Atrachus flagella example is Xylella, Peritrachus is Irvinia, Cephalotrachus is Pseudomonas fluorescens. Okay, a natural genetic engineer. There is a bacteria called natural genetic engineer, or farmer's friend also we can say. That is Agrobacterium tumefaciens, right? And the next one is a common scab of potato can be controlled by the reducing pH to 5.2 by the application of. So for reducing the soil pH, what we apply to the soil, generally we can apply zinc sulfate, ZnSO4 at the rate of 900 kilograms of soil to reduce the soil pH. If you want to increase the soil pH, we can apply the lime at the rate of 2.5 tons per hectare. The next one is first seed health testing laboratory is established at Copenhagen, Denmark. So Copenhagen is that the capital for Denmark, where the first seed health testing laboratory is established. First official seed pathologist Louis Dyer. Louis Dyer in probably 1938 is the first official seed pathologist. This is about bacteria, I mean flagellum, Atrichus no flagella, Xylella, Monotrichus xanthomonas, Amphitrichus pseudomonas, Peritrichus irvinia, Pantovia, Escherichia coli, Clostridium, Lopotrichus spirulina, Cephalotrichus pseudomonas fluorescens, Circumtracheal, Agrobacterium tumefaciens. There are the representation photos. For further information, students can refer my book, A Vision into Plant Pathology, a complete student version. For further doubts, clarification and guidance, students can reach us at www.geekyresearcher.com. Stay geeky and stay tuned. We are Team Geeky Researchers.